Basic life history information from animal species is vital to conservation efforts. When it comes to avian species, understanding things like their dietary composition is vital and of utmost importance when it comes to their conservation. But let's now introduce our key player of this story, the federally endangered red-cockaded woodpecker, or RCW for short. The RCW itself is a federally endangered cooperatively breeding species. This species can lay anywhere from 1 to 4 eggs, and they rarely produce a second brood. And this species also thrives in fire-dependent communities, specifically the longleaf pine ecosystem. RCW population sizes have dramatically increased in recent years, resulting in unprecedented population densities. Within recovered populations, scientists have recently noticed increased rates of early and late partial brood loss, and this partial brood loss could have something to do with RCW diets. So what is known about RCW diets? RCWs themselves rely on a specialized prey community, which includes prey items like wood roaches, wood borer larvae, and ants, to name a few. However, the diversity of their prey base is largely unknown. Additionally, RCW's prey resources may be impacted by a changing climate. Elevated precipitation and temperatures can cause phenological mismatches in which birds rear broods at a time that does not coincide with maximal food abundance, which therefore could be having and causing this impact of partial brood loss. Even with using traditional methods, there's still very limited knowledge with RCW prey. So therefore we ask the question, is there a better way to measure RCW prey? By using an intense genetics analysis, there is a way to sample RCW prey using RCW fecal sacs. Taking the collected samples, the DNA will be extracted using the QIAGEN Power Soil DNA Kit. This will then be barcoded with each DNA sample individually to allow for HTAS and will include forward and reverse ANML primers as well as negative extraction controls for both extraction methods and use high throughput amplicon sequencing. Then with using the high throughput amplicon sequencing, single copy mock community consisting of DNA from insects that we expect to find will be used and extracted to analyze the presence of these various diet items. The end results will help us identify all possible prey species of RCWs. And the ones in red are actually pest species. So in conclusion, using this very intense genetics approach with high amplicon sequencing and bioinformatics shows us that RCWs consume a wide and variable diversity of arthropod taxa, including pest species. Bioinformatics, therefore, is a useful and powerful tool. By using this tool to look at insect richness at various sites that are experiencing partial brood loss, will help us get to the bottom of a possible connection with insect richness.